All right. All right, so we're going to begin. All right, Freya, do you see the screen and everything? Freya, can you hear me? All right. Okay. All right. So let's begin with the. So we're going to be doing, DNA replication, right? So right off the bat, so Anas, so what do you think that means? DNA replication. What do you think that could mean? Like in your own words, what do you think that could
Hi,
mean?
brother.
Copying genes of another person.
So copying, right? Replication means to what? To copy, right?
Yeah.
So whenever we hear some kind of word, like a scientific word or something, right, we should always try to go back to, like regular language and try to figure out, like, okay, replication. Try to figure out logically what could this mean, right? Then you study it, and then it helps you to remember, like, what this term means because you understand, like, how it logically fits together, right? All right.
Yeah.
So the example that they're just giving here in the beginning, right? So for example, you have Morse code, right? So Freya, have you ever heard of Morse code? All right. Hello. All right. So what is Morse code? Can you describe it in your own words? Like, what is Morse code?
It's like a code that has different types. It's like not letters, but like it has
Mm-hmm.
lines and dots.
Right. Exactly. Right. So you have, like, you see little dots and lines, right? So the different dots and lines, they code for the letter, right? So all you have is, like, you see how you have, so dot and line. How many different things is that? That's just two things, right? You have dot and line, but you put them in different numbers, different orders, arrangements, combinations,
And then you
and you make 26
letters.
different letters. You see that?
Mm-hmm.
So that's kind of how DNA works, right? So does anyone know how many different types of nitrogenous bases do we have in DNA? Anyone remember? We're going to talk about it, but, like, anyone remember
How many different types of night? 4, right?
Basically 4, right? So you have, like,
Mhm.
A, G, C, and T, right? So you have 4 of these.
Adenine, guanine, cytosine, and thymine, right, brother?
And thymine, right?
Something like that.
So these
Yes.
4 things, right? You only have 4 different things, okay? But using these 4 things, you can code for, like, something like more than 20 different kinds of amino acids. So you understand how this is the same kind of concept, right? We're only starting with, like, 4 different things. But you put them in all different combinations, different orders, and each of those is like a different code, right? So in DNA, you take 3 of these things, right? You take 3 of these bases, and that codes for an amino acid. So for example, you could take A, G, C. Then another one could be like T, A, C, T, A, G. You see all these different combinations, right? So you can make a lot more than just 4 different amino acids, all right?
Yeah.
All right. So first, we're going to look at a little bit of history. So we already talked about Mendel, right? So he did some work on genetics. We talked about the Punnett squared, all this stuff, right? So this was like the basics of. So in this person's time, there was no, like, microscopes. There was no, like, they didn't know, like, much about DNA, or they didn't know what DNA was. So he didn't know anything about DNA or chromosomes or anything like that, right? He didn't know about those kinds of detailed things in the microscope. But he just noticed, like, mathematical patterns. Does that make sense? Right? So he just noticed mathematical patterns. So all the stuff we talked about, right? We know now that there's like 2 different chromosomes, one from the mother, one from the father, stuff like that. But he didn't know any of that stuff. He just noticed, like, different patterns that arrived. All right? All right? So this person over here, Walter Sutton, so he discovered that chromosomes carry a cell's genetic information, right? So we talked about all those different kinds of chromosomes, right? Remember, we've talked about different chromosomes, right? So overall, Anas, in a human being, how many total chromosomes are there?
My brother?
So how many total chromosomes are there in a human being?
I don't remember.
You remember? Yusuf, what do you think?
Come on this. 23 pairs, brother.
So 23 pairs, right? So 23 times 2, 46, right? So I think these were like supposedly some early drawings that this person made. So you notice, right? So when cells divide, right, all the DNA comes together into these nicely formed chromosomes, these kind of like blob type shapes. So he observed some of these and he drew them, and so this was the first trying to figure out figuring out that there was such a thing as chromosomes, right? This person over here, he discovered the linear arrangement of genes on a fruit fly's chromosomes. So what does that mean, the linear arrangement of genes? So linear means by a line, in a line, right? So that means that there's the arrangement of genes. So what is a gene? Fariha, do you remember, like we talked about one time, what is a gene? Do you remember what this word is, genes? So, so we have DNA, right? We have it's like very, very, very long, okay? But so the DNA is divided into small, small pieces. And each small piece, right? I mean, they're all linked together, right? They're all, physically, they're all together. But in terms of its function, it's like made up of like different, different, groups of, different, sets of DNA base pairs, right? So when each piece of the DNA, right, codes for like one protein, right? It's a little bit more complicated than that, but you can kind of think of it like each different segment codes for a protein. The next set codes for another protein. The next set codes for another protein. So he discovered that on DNA, right, that, if you look at the DNA, that, all the different genes that code for proteins are arranged in a line. So let's kind of take a look at like a little picture of it over here. So you can kind of see here on the chromosome, right? Let's take a look at this chromosome over here on the right. So you notice that they shaded different portions, right? You see like on the bottom, they shaded like a little portion. You go up, there's another portion that's shaded. You go up, there's another portion that's shaded, and so on, right? So now here they're only showing like 6 or 7 on the chromosome, but there's going to be like millions and millions and millions of these different genes. Maybe not millions, maybe probably like thousands or hundreds of thousands on each chromosome. So each of these little pieces codes for like a different kind of protein. Does that make sense, right? So each on this chromosome, like this portion over here, codes for one protein. You go up, this gene over here codes for a different protein. You go up, this other gene over here, another protein. You go up, another gene, another protein, like that. So you see how they're arranged in kind of like a line, right? Linear fashion. They're linearly arranged, right? So this person came up with this idea that these things are arranged in a line. All these genes in a line. And so each gene is like kind of like one protein, basically.
Each one of those lines inside the helical helix, right?
Right. Each of these little lines, each of these genes is like one protein. This, gene 2, is another protein.
Wait. Each line inside the DNA helix or whatever, right? Each little horizontal line?
No, not horizontal line. So like from here to here, like this portion here is like one gene.
Okay, but how do they know from that portion to that portion? Like, how do they know that?
Oh, yes. So we're gonna talk a little bit about that. So there's different. So within here, there's like special sequences that are recognized as called like promoter regions. So there's like this thing called a polymerase. It's like a protein. It's going on the DNA, reading, and when it reads certain sequences, it knows, okay, I better start copying or start making some mRNA right now. So there's different sequences that when the protein recognizes that order, it knows, okay, this is where I should start copying things. So we'll look at another picture of that later.
Okay.
All right. This person over here, Griffith, he found that bacterial cells contain a molecule that transfers genetic material from one cell to another. So this person was looking at bacteria. All right. So in bacteria, the DNA is like a lot simpler than in human beings, right? So it's a lot simpler, right? So in this person, what he did was he took some different kinds of bacteria, right? So he looked over here. So this is a very interesting kind of experiment, right? So he took this strain of bacteria over here. It's called living S strain. virulent, right? So what does virulent mean? Freeha, do you know what virulent versus non-virulent means?
No.
Any ideas, right? So basically, it basically kind of means like it's like infective. It's like infectious. It's it's dangerous, right?
Mhm.
So if you put this bacteria into this mouse, the mouse gets sick and dies, right?
Mhm.
Now you take this other kind of bacteria over here, right? Now this bacteria over here, right? This is also
Yeah.
alive, but it's like a non-virulent strain. This one is not really like too dangerous to the mouse. So when they inject it, like nothing really happens to the mouse. Okay.
Mhm.
Now, they take the you see this this dangerous bacteria over here, the S strain. So what they do is they take this bacteria and then they basically kill it. All right?
Mhm.
They heat it up, so the bacteria is still there. There's like DNA floating around in there, whatever, but the bacteria itself is dead.
This is just dead.
Right? So they take that in the third one and they inject it in the mouse, and so the mouse lives. Right? Now, they took some of this dangerous bacteria that they killed, but they combined it with this bacteria here that's not dangerous, right?
Mhm.
So they took this bacteria that was killed, right? That's dangerous, but it's killed. And they took this one here, the non-dangerous one, and they combined them together in like a test tube. So they let it sit there for a little bit and they mixed it, right? Now, neither of these individually, you can see, right, 2 and 3, neither of these individually are going to do anything to the mouse. But when they mixed it together, all of a sudden the mouse got sick and died. So what does this mean? Anyone have any ideas? Like, what does this, what did he, figure out from this? What does this experiment show?
He
You can't
originally
mix two genes from different people.
You can't mix genes from different people, okay? Yusuf, what do you think?
he found out that so he originally found out that DNA replication exists or something like that from this thing, right?
Kind of.
I think whether he found out that like the OK genes, like the non-virulent ones, or the bacteria, the non-virulent bacteria, it basically like copied somehow the structure of the virulent bacteria and it replicated it.
All right, Freeha, what do you think?
So I figured out.
Okay, before that, I have a question. What's the living S string?
So S and R are just like random names. S is just like the dangerous bacteria. R is like the non-dangerous bacteria. So it doesn't matter that it's called, we can call it X and Y, right? It doesn't matter. It's just S is the dangerous one, R is the non-dangerous one. So I think,
So S wouldn't mix with anything to,
all right, so over
so S itself is like dangerous, right?
is dangerous, right? Now in the third one, they took the S, but they killed it with heat, right? They heated it up, so they killed it, so now it doesn't kill the mouse. But if you mix this one that was heated and destroyed with this non-dangerous bacteria, all of a sudden that combination killed the mouse. So,
Combination is dangerous to the
so Yusuf was pretty close. So here's what this kind of demonstrates, right? You see in this mix over here on the right, all the way on the right, right? So you have the non-dangerous bacteria, and then you have the killed dangerous bacteria, right? So now the dangerous bacteria, it's dead, right? So it shouldn't harm the mouse. But what happened was that you see the non-dangerous bacteria, right? What it did is that it took up the DNA from the dead dangerous bacteria.
So I was right, brother.
Right? So the dangerous bacteria is dead, but its DNA is floating around in the test tube. And so what happened is that the non-dangerous bacteria was able to take that in and use that DNA. And so now it can make the proteins and stuff that are needed to invade the mouse and kill the mouse and make the mouse sick. So this is something that bacteria do. So most like human cells and mammals and stuff like that, they don't do this kind of thing. But what this shows is that the genetic material, like the material that tells the cell what to do, right? there's something there that can be transferred from one thing to the other. Does that kind of make sense?
Yeah.
So I don't.
Like,
I
can
don't
you repeat that one more time, brother?
Yeah, so this just means that the material, the genetic material, so again, he didn't know about DNA specifically, but the genetic material that tells the cell what to do, there's like something that you can isolate and take out and put into another cell.
Uh-huh. And so that's what he figured. So that's what he kind of figured out.
That's
what
what he
like you could deduct from this.
That there's some kind of written instructions somewhere that we can take out and then we can send to another cell. All right, so this person. All right, so let's look at this one. This is called Chargaff's rules. Right. So we looked at different the different base pairs, right? So remember, there's 4 different base pairs in the DNA, right? So we talked about there's adenine, guanine, thymine, and cytosine, right? Now A and G, you notice A and G, these are a little bit bigger in size, right? You see that? A and G, there's a hexagon and a pentagon attached to it. So A and G are going to be like a little bit bigger in size. Now T and C, right, are basically just a hexagon. They're basically like smaller in size than the A and G. So what Chargaff's rules are, what this person found out is that you see A always binds with T, and G always binds with C. So you look over here on this DNA, right? Remember, DNA has 2 strands. You see the left strand over here going down here, and then you have the right strand over here on this side. So each of those has like a base pair, right? So for example, this one up here, right? The A over here is binding with the T from here, right? So A and T.
Wait. Brother, what are these called? These 4, are these what?
These are called nitrogenous bases.
Nitrogenous bases.
Right. And look at the next one, right? G and C. So again, G and C. So it's always A and T, G and C. There's never like A and C or A and G, nothing like that. Always A and T and G and C. All right, that's always what it is. So what does that mean, right? It means that let's suppose we know, okay? Like you see the strand on the left, T, C, A, G, right? T, C, A, G, right? And let's suppose we only have this one on the left, right? Right? How could I reconstruct the one on the right? Let's suppose I only know the one on the left. How could I reconstruct the one on the right? Freya.
Yes.
Right, let's suppose you know that this one is T, right? And this one was missing. Right, so this one was T. So how could you figure out what this one was over here?
It would be the shorter.
So it would have to be A, right? Remember T and A always go together, right? That means that if you have T over here, you know that the other one that's attached to it has to be A. It cannot be anything else. Right? You see this one over here? C, remember we said G and C always go together. G and C always go together.
There's no other compromise. Like it's never anything else.
Yeah, never anything else. Unless there's a mistake. There could be a mistake. That's a different thing. But like regularly, there's no. It couldn't.
But
be
if there's a mistake, that means it's a genetic disorder, like
so
disease
there,
or something like that.
there's like DNA. There's like mistakes, and then that could cause either that's fixed later on, or maybe it just gets destroyed, or it causes cancer or something like that. But that's basically like a problem. That's not what it's supposed to be.
It's not normal.
Right.
All right.
So you have C. If you have C here, remember C always goes with G. Right. C and G always go together. So if you have one, that means it's always going to be the other. So if you have this strand on the left, right, and you don't know what is the strand on the right, you could reconstruct it. Now, if you have T, you know, okay, I got to put an A with it. If there's a G, okay, I got to put a C with it. If there's a C, I got to put a G with it. Right? So if we have the strand on the left and we don't know the one on the right, we can reconstruct it if we know that, this rule called Chargaff's rule. Okay? So we'll do there's an exercise later about this. All right, so this person over here, Rosalind Franklin, was a scientist who used X-rays to look at molecules. So like the basic idea is just that, there's this process called X-ray diffraction that they talk about here. So if you take a molecule, right, and you send it, and it has a certain geometry, and you send an X-ray beam at it, the X-ray beams will split apart and they'll like go off in different directions, right? And then you put like some kind of paper behind it to read and look at that pattern. And based on that pattern, they know how to figure out what is the shape of the molecule. Not usually like super complex shapes, but like simple shapes, right? You throw an X-ray beam through it, the way that it splits and the pattern that it forms on the wall. Based on that, you can tell what is the shape of the DNA. So I don't know how exactly so this is apparently like the pattern. You see this picture here? This is apparently like the pattern that they got. And based on that, they figured out that DNA is a helix. So helix is something that curves around, right? DNA kind of like spins around, right? It's kind of like this curved helix that kind of spins around. So based on this X-ray picture, they figured out that DNA is a helix.
Oh.
So and I think this is like a famous picture that you might see in a lot of places. So this is the X-ray picture that they found. So based on this picture, they figured out that DNA is like a double helix kind of shape. And so these were the people who, so I think Watson and Crick, they were famous scientists. They worked with this other scientist, Franklin. And so they figured out like more, some more details about the shape of DNA in like a little bit more detail. So right over here, this is the shape of DNA. So these are these scientists on the left. They made like this model of the DNA, right? So take a look at this, right? So Freya, why do they call this a double helix? Look at this picture over here. Why do you think they call this a double helix? All right, so Freya, just keep.
Because there's a double strand.
Okay, so there's two strands. Okay, why is it called a helix?
I don't know.
So Yusuf, why do you think this is called a helix? What does that mean?
Because, brother,
Helix
it has-
or helical shape?
It has those rotating lines around, and in the middle there's those horizontal lines, and that's the helical
Right,
shape.
so a
There's like
helix
two,
is something that kind of twists around. Okay.
Yeah.
So a helix is something that kind of twists around. Okay. So Freya, let's go back to it again. So a helix is something that twists around. So describe that shape over here. What does this have to do with a helix?
There's two strands twisting around.
Right. So if you look,
Over each other.
right, if you look at the picture, right, one of the strands is like going in the back and then coming back in the front, then going in the back and coming back in the front again, right? So they're twisting around each other. So one is in the back, the other's in the front, then it twists around, right? So that's why it's called a double helix, right? And so you notice these like little letters in here, right? This is what we're talking about. A and T always go together. A and T, G and C, right? So you can kind of see some of those letters in there. You see that? A and T, A and T, G and C, G and C, right? So this is the structure of DNA. This is what it looks like, right? And then this over here at the top, this is kind of like, so the two strands, they can come together, they can come apart. So A and T, right, and G and C, they're not held together by like super strong forces. That means that, you can break them apart and unwind the two pieces of the DNA, right? You can unwind the two pieces of the DNA. Right? Now, these two people, they basically mapped the human genome. So what does that mean? That means that, the whole DNA.
Hey, brother, what is the genome again? I still don't understand.
So
Genome is like the makeup of the.
yeah, so
human?
the
Genetic?
genome is the entire sequence of the DNA from start to finish. Like, A, T, G, C, A, C,
But like,
A,
it's
A.
not always the same thing, is it? Like
No, no,
people
right,
have
so,
different.
right. So it's not the same thing. So that's why they mapped the whole thing. So
What
whatever.
do you mean they map? How do they
So,
map?
so look at the bottom over here. You see the sequence at the bottom,
Yeah.
right?
Mhm.
So from start to finish, they mapped the whole. Mapping means that they figured out what are all the little base pairs, A, T, C, in what order.
Mhm.
So they figured out the entire order of the entire human genome, all the chromosomes, from chromosome, all 46 chromosomes. They figured out from start to finish, what is the order of the base pairs, C, G, A, T, the whole thing, billions and billions and whatever, trillions, whatever it is, of base pairs. They figured it out. Right?
Can you repeat that?
Yeah, so they figured out all of the base pairs of right? So there's 46 chromosomes, right? So each chromosome, right? If you start at the beginning of the DNA and just start reading it, look at all the base pairs, right? A, T, A, G, A, C, like that, right? Right? From start to finish, they figured out, and it's written down somewhere, like the entire, like all of the base
That's
pairs.
That's a long
Yeah, so,
thing, right? Because
yeah,
there's a lot of base pairs.
right. So it's gonna be pretty long. So they figured out, and they, like what is the order of the entire human genome, all of it. Okay. All right. So let's take a look at some of the structure of the DNA, right? So we talked a little bit about nucleotides before, right? So let's take a look at some of these pictures. All right. So we're going to take a look at a nucleotide. All right. So first of all, you see over here, these are the two. So DNA, we said double helix, right? Double meaning two parts. So we have the left part over here, and then we have the right part over here, right? See the left part over here, and the right part over here, right? And notice again, right? AT always goes together. AT, GC, CG, AT, right? So A and T are always together. G and C are always together, right?
But like there's like a certain amount of like base pairs for each what's it called, for each amino acid.
Right. So three of these. For example, GCA could be one.
Oh, like so three of these base pairs, so GCA.
Right. So basically,
But
three
like,
of,
how would you?
yeah. So
Oh, because you can
we're
identify
talking
either
about-
way.
We're going
'cause
to talk
there
about
was-
those details a little bit later. Right.
Oh.
So over here, like, what is a nucleotide, right? So you notice here, there's kind of like a pattern, right? So you have like a base pair, like A, G, C, or T, whatever, right? For example, let's take this one, A, right? So we have this nitrogenous base. This thing just by itself is called a nitrogenous base. Now the big ones, remember the big ones, A and G? These are like the ones that are big in size. See that?
They're called purines, right?
Those are called purines, right? Correct. So these are like a hexagon and a pentagon put together, called purines. The smaller ones, like T and C, you notice these are smaller in size, right? These are called pyrimidines, right? So notice you always have one purine and one pyrimidine, right? So if you have like two purines, that would be too big, because purine takes up more space. So there's gonna be like a little bulge in the DNA over here, and so they're gonna have to fix it later, right? But so you see, okay, so you take one of these base pairs, one of these nitrogenous bases, like the A. You take this deoxyribose sugar. You see, this is a sugar over here. We talked about the sugars before. And
Okay, so.
and we take this phosphate group here, PO4. You see this P and 4.
Oh, so this is the hydrogen bond, right? This is a
Wait.
hydrogen
No,
bond.
just a second. So these three together, right? The phosphate, the sugar, and the nitrogenous base pair. These three put together, this combination, this is called a nucleotide.
Wait,
Okay,
brother,
it's the hydrogen bond. I see. Right.
what do you call it? Isn't the pair for A supposed to be U?
No, so U is for RNA. We're going to talk about that later.
Oh yeah,
So,
the
okay.
RNA
Okay, good.
strand.
So in DNA, you have these ones, A, C, G, and T. So we're going to talk about RNA a little bit later.
Okay,
Right.
so just to clear up, brother. So the pentagons on the either side of the bases nitrogenous bases. Those are deoxyribose.
Right.
Oh, sorry, not deoxyribose. Sorry. What is this?
Yeah,
What is
right,
supposed to be?
right, right,
Hydrogen
right.
rock.
Urine.
Yeah, you see these blue pentagons.
Mhm.
This is deoxyribose. It's a kind of sugar.
Oh, okay. Deoxyribose. Yeah, ribose, right?
Ribose, right? All right. So now over here, you see these little red things over here. These little red dots. What is these red dots showing?
The hydrogen ion bond.
Close, right? Hydrogen bonds, right? So these are hydrogen bonds.
Mhm.
So these two strands of the DNA, the left part and the right part, they're held together by all of these little hydrogen bonds. Hydrogen bonds here, you go up, more hydrogen bonds, right? So hydrogen bonds, right, are they stronger or weaker than covalent bonds?
Weaker.
So these are pretty weak bonds, right? But it's just that you have so many of them, right, along the whole entire length, right? Right? Every
Makes it
base
worse.
pair, A to T, GC, CG, there's like all of these, right? So you put them all together, and overall it keeps the two sides of the DNA together. Does that make sense, right? So it's pretty weak,
But the covalent bonds are still stronger, right?
right? Covalent bonds are so strong, right? So these bonds are weak. That's why you can take them apart. That's why you see the left strand and the right strand, you can take those apart. However, there's like so many of these that, most of the time, unless you physically like take it apart, most of the time the DNA, stays together. All right?
And the deoxyribose are connected like vertically, basically, right? Through what's it called? Covalent bonds between the phosphate groups.
Right. So these are all, so pretty much everything else here is a covalent bond.
Right. So basically, this deoxyribose is connected to this phosphate group, and this phosphate group is connected to the other deoxyribose molecule, right?
Right.
It's kind of like a
yeah,
chain.
Correct. So so all these other bonds, everything else here, covalent bond.
Mm-hmm.
All right, so nitrogenous bases, right? So we talked a little bit about this, right? So you have purines, right? The large ones. You have guanine and adenine. These are the larger ones. And then you have cytosine and thymine in DNA, right? Right. Now, this one over here, you see this one called uracil, okay? This one over here, U, right, is basically the same as T, okay? So U is basically the same as T, except it's only an RNA. So in DNA, you have T, okay? And
Okay,
its
but in
place,
RNA, you have R.
You have U,
U.
okay? So T and U are basically like interchangeable. I mean, they're basically the same thing, except just in DNA, you have T, in RNA, you have U. All right? All right, and this is just like another example that's kind of showing, right, the different how DNA kind of looks, right? So again, it's a double helix, right, because you have two pieces, right, double, and it's a helix because it's like twisting around, twisting, helix, right? So notice again, you see the base pairs, right? It says on the top, it says A, C, G, T, A, C, T, right? So let's suppose we had only those ones, right? Right? We can figure out the ones on the bottom. Because notice A and T go together. So if I have A, I know the bottom one is T. If I just had this C, I know the next one is G. If I know that this one is G, I know this one has to be C.
Wait. Okay, yeah, but you don't know the one that's above or below it, right? Like, for example, I have a bond that's T and A, right? Thymine and adenine. But I don't know the next one is C, because the one before is thymine. Then I don't know the next one is cytosine.
Right. Here's what I'm saying. Here's what I'm saying. I'm saying if you have only one strand, right, and the other strand is missing, okay, you can recreate the other strand.
Yeah, you can recreate it because you know
Right?
what
Because
bonds with what.
each of these base pairs only forms a bond with one other. Like
One other base.
right?
One other
Because T
nitrogen.
always goes with A. It never goes with anything else. If I have a T here, I know that this one here has to be A. Right?
Right,
do you understand what I'm saying?
Mhm.
Right? If I know that this is C, right, I know that this one has to be G, right, and so on, right? So that means that if these letters were missing, that you would be able to fill them in, right? Right. So hydrogen bonding, we already talked a little bit about hydrogen bonding, right? So this picture is just showing like a little bit more detail, the hydrogen bonding going on. You see the dotted lines, right? You see these
Yeah.
little dotted lines, right? Those are hydrogen bonds, right? Because so those are a little bit weaker than covalent bonds. That's why they just put little dots. Okay. Again, A and T always go together. G and C, right?
Mhm.
Right. Sorry. Okay. All right. So we have base pairing, right? So we talked about this again, right? G always goes with C, A always goes with T, right? So this is called base pairs, right? So base meaning nitrogen's base, pairs meaning two, right? So each of these, G, like for example, G and C, right? We call this a base pair, right? Or like a pair, right? Pair of bases, right? Base pair, pair of bases, two bases, right? All right. And again, this is kind of showing the same kind of picture, right? C and G. Now here's another thing, right? You notice between C and G, right?
Yeah.
How many hydrogen bonds are between C and G?
3,
3, right? How many between A and T?
2.
2. So which of these base pairs is held together a little bit stronger than the other?
C and G.
C and
C
G,
and
right?
G.
Right? You see how like C and G have 3 hydrogen bonds. So this is going to be held together like slightly more stronger than A and T are held together.
Is
Just
that
like-
always the case? C and G always has 3 hydrogen bonds, and T
Yeah,
and A
yeah.
always have 2.
Yeah, unless there's some mistake or some problem, right?
Mhm.
But it always has 3 hydrogen bonds. All right. So now that we talked about those concepts, right? How does DNA replication work? Okay. All right. So let's take a look here. So we have the original DNA here, right? Let's say we have this is the original DNA here, all right? Now, these hydrogen bonds in the middle that we talked about, they're broken apart, right? There's an enzyme that comes around and kind of tears them apart, kind of takes it apart, all right?
Mhm.
So now you see the DNA strands are no longer together. See that? Right. So now you've taken the 2 strands apart. And then what happens? Each strand, right, forms its own copy, right? So in the beginning we had these 2 green strands all together, right? Left and right. We break them apart into this left and this right section. Now this left one goes up top here and forms its own copy to form the other strand.
Mhm.
This
Oh,
one
it
on
can
the
form
right
the
here-
other copy because the base pairs are always the same, so it knows
Exactly,
exactly.
right? So Yusuf, explain. So how is this possible? How can this, DNA from the right go over here and just kind of make its own copy? How can that happen?
Because the base pairs are always like adenine can only bond with thymine, and cytosine can only bond with guanine, so it automatically knows what to make on the other side for one strand.
Correct, right? So you notice that if this was a T, right? You see this one is a T here at the top. You know that the other one can only be A.
Mhm.
And if you go back to the original, you can see, oh yeah, that one was also A. So you see the fact that these rules exist, that A can always go with T, can only go with T. G can only go with C. So if you take them apart, well, now you could reconstruct another DNA piece, another DNA strand for each one, and now all of a sudden, now you have 2 pieces, right? So look at the end, right? DNA has been replicated. Instead of 1 piece of DNA, now you have 2 pieces. See that?
Mhm.
Right? So this is the basic concept, right? Because A and T can only go together and G and C can only go together, right? You can take them apart and you use each of those to make another copy for each one. And now, all of a sudden, now you have 2 pieces of DNA. So DNA replication, right? So you notice each of these, the 2 strands of DNA, the left and the right, right? Notice how they go together, right? A and T, G and C, they're matched up in that way. So these 2 strands, right? The 2 strands of DNA that go together are called complementary.
Mhm.
See that? That's the word, complementary. So when you have this double helix DNA, the left strand and the right strand, those are complementary strands of DNA. All right?
Mhm.
So let's look at, I think I put some more pictures, right? right? So notice over here, right? You have A, A, T, T, G, G, C, C.
Mhm.
So based on this, what do? We know that T goes with A, T goes with A goes with T, and so on. So for example, if I have this question here, what is the complementary strand of DNA, in the black? Right? Freya, how would you figure out that answer? Hmm?
Like with A, T goes with A and A goes with T, G goes with C, so we know the put them like that.
Yeah, so explain for this one. Tell me for this specific example, explain how we got this answer.
So they gave us A, T, T, C, C, G. So A pairs with T, so that's gonna be the first. And T pairs with A, T pairs with A, C pairs with G, and G pairs with C.
All right, good, right? So if we have one strand, right, then we know how to make the other strand. And so this is how DNA replicates. This concept is very important for DNA replication. Now this is showing a little bit more in detail, right? So for example, this one on the right, right? So we're starting here on the right. We're starting here on the right. And so we have this double-stranded DNA. Now this one over here, you see this thing called helicase? This is kind of a protein, right?
Mhm.
There's a protein called helicase. And this helicase kind of like unwinds it and takes it apart, right? It's one by one, it breaks those hydrogen bonds, and takes it apart, takes apart the DNA, right? So this helicase is kind of unwinding it over here. So then the one on the top here, right? These additional fragments of DNA come and lodge in over here, right? There's this protein called, polymerase.
Yeah.
Right.
Enzyme, right?
Yeah, enzyme, right? So this is an enzyme that helps the proper, base pair to attach. So if there was a C, it makes sure that there's a G. If there's a T, it makes sure that an A comes along and attaches like that, right? So you have these proteins called polymerase. It's not called RNA polymerase here. I don't think this is correct. This is called DNA polymerase. So DNA polymerase is this protein that attaches here. And so in the end, right, you have two pieces of DNA. You see one on the top here
Brother,
and one on the bottom.
in the
Just so that-
RNA
so that- this is
strand
called
of like
DNA replication.
Hold up.
All right, go ahead.
like in the other RNA strand, now A would be with you, right? In the second, like after they divide.
No, only if you're making RNA. But here we're making two pieces of DNA here, right?
Wait. So the enzyme polymerase makes the complementary strands, or
Yeah,
no?
it helps to basically put it all together.
Put the two strands together, right?
Yeah.
Right.
So it's called DNA polymerase. Right. All right, so let's talk about RNA and DNA. So far we're just talking about DNA, right? DNA replication. If you have one piece of DNA and we want to make two. So first of all, Anas, why is DNA replication important? Like when is this used?
I don't know.
Not sure. So why?
Kind of like the nucleus and stuff.
But so why would we want to double the amount of DNA, right?
DNA replication, brother. So when you make new cells.
When you're making new cells, right? Remember the things we were talking about, mitosis and meiosis, right?
Yeah,
We wanna
that's
take-
what happens in those
Right?
processes.
We want to take
right?
one cell and make like 2 cells or 4 cells or whatever, right?
Yes.
So that's when you want to.
But in the case of meiosis, it's different, right, brother?
Yeah, correct. So meiosis is a little bit different, right? So mitosis is the simple example, right? So in mitosis, we want to double the amount of DNA so that each new cell gets its own full copy, right? So that's why that's important. All right. So we have DNA, and then we also have RNA, right? So RNA, there's a couple of differences, right? So first of all, we're gonna look at some pictures, but let's take a look at these 3 things. So first of all, remember DNA is usually 2 strands, right? Double helix. RNA is usually only 1 strand. All right. Now remember the sugar in DNA. Freya, what was the sugar in DNA? Do you remember what was the name of that sugar in DNA?
The sugar name in
Ribos.
Close.
Deoxyribose.
Right. So in DNA, deoxyribose, right? So you can think of it as, both of them start with the letter D, right? Deoxy DNA, right? So RNA starts with R, and so its sugar is ribose, right? So the two R's go together, the two D's go together. Okay, so that's how you can think about it, right? So RNA has a slightly different sugar. It's called ribose, not deoxyribose. And third, remember we talked about this one. Instead of T, we're going to have U in RNA. All right. So let's take a look at some pictures of what's the difference, right? Yeah, we're going to talk about what it does in just a second, right? So first, let's talk about what it looks like how it looks like different. So remember, DNA. Look at this. DNA has two strands to make this helix like this, right? 2 strands.
Yes.
RNA, usually, it's just one, right? Sometimes it's 2 in like certain viruses and stuff like that, but most of the time, it's just as 1 strand.
Oh, RNA is just one strand?
1 strand, usually 1 strand.
Then how does it have the base pairs?
Yeah, so we're going to talk about how it copies it. So it gets its base pairs from DNA. It copies it from DNA. That's how it gets it. All right, we're going to look at those pictures a little bit later.
All right.
DNA has deoxyribose with a D, right? RNA has ribose. And then, instead of thymine over here in the pink, it has uracil. So uracil functions the same as thymine, right? So there's no real difference in how it functions, right? Just like T goes with A, so now U is going to go with A. Does that make sense, right?
It probably has a different makeup though, like a slightly different makeup or
Right,
something.
right. So you can look at the structure; it's slightly different, right?
Oh yeah, that one has 3 hydrogen, right? And 1 carbon, and this one doesn't have any hydrogen or carbon.
Right. So it basically functions in the same way. It just has a different structure. So T goes with A usually, right? So now U is going to go with A, right? So those are basically the 3 main differences, right? And this picture is kind of showing the same, kind of thing over here, right? So basically, they're all the same except for uracil, U instead of T. And then RNA is 1 strand, DNA is double strand. All right. So there's different kinds of RNA, right? So there's messenger RNA, ribosomal RNA, and transfer RNA. So let's take a look at the different kinds. So you see messenger RNA, right? So what do you think this means, Freya, messenger RNA? Why is it called messenger RNA?
It gives instructions or it sends message.
Yeah, so, right. So basically, the DNA, right? The DNA is usually like stuck inside the nucleus, right? It's just the DNA is always, so the DNA is just stuck inside the nucleus, right? So how does the DNA communicate with the outside of the nucleus? Well, it sends a message through messenger RNA. So the messenger RNA is basically sort of copied, like a certain portion is copied from the DNA, and then it floats outside. It can go do certain other kinds of things to make proteins. So messenger RNA, right, is copied inside of the nucleus, then goes outside in order to make proteins.
So basically, it's copied from DNA inside of a nucleus, and then it goes to deliver that, whatever it is, it copied to like some other part for some purpose.
Right. Now, ribosomal RNA. Remember, Anas, what do ribosomes do? Remember what ribosomes do?
I don't, brother.
You remember? So ribosomes, Yusuf, do you remember what do ribosomes do in the cell?
Ribosomes, I believe, brother. I'm not sure though, but I think they make proteins, right, brother?
They make proteins, right? So the ribosomes, they're attached to the rough ER, or they're floating around in the cell sometimes, right? So the ribosomes, they have RNA within them, right? So within them, they have RNA, right? Now, you also have something called transfer RNA. Transfer RNA. So transfer RNA, right? You see this little blue dot at the top?
Brother, what does the RNA in ribosomes do?
So the RNA and ribosomes, they probably attach to the messenger. I'm not sure, I don't remember exactly what they do. They probably help guide like the messenger RNA through and stuff like that.
But then, like, what did you mention about the ribosomal RNA? Because I didn't hear.
Well, I just mentioned that this is part of the RNA, part of the ribosomes. That's all I mentioned.
So okay.
And then let's look at transfer RNA. So transfer RNA transfers something, right? So you notice this blue dot at the top. Anyone know like what is this blue dot at the top here?
I don't know.
So this is an amino acid. So every transfer RNA has a different kind of amino acid. So notice here we were talking about, you know, if you take three of these bases over here, right? For every three bases, there's basically it codes for one kind of amino acid. All right. So we're going to take a look at how all this is put together. But
Wait,
right
can
now,
you
just
repeat that,
remember,
brother?
you know, kind of the general structures of these things. All right.
Anyway, brother, can you repeat the transfer RNA one more time? Sorry.
Yeah, so transfer RNA basically holds amino acids, and this is involved in making proteins.
It's not involved in making proteins?
No, it is.
It is involved.
Yeah, it is involved. All right. So let's take a look at these two words over here, transcription and translation. All right. So at the top over here, we have DNA, right? See this double-stranded DNA? So now a portion of the DNA is copied into RNA, usually messenger RNA. Remember we talked about messenger RNA, mRNA.
Yeah.
Messenger RNA, or they also call it M for messenger, mRNA.
Mm-hmm.
So DNA is copied into mRNA, right? So this process here, where DNA is turned into RNA, this process is called transcription. That's the name of this process, transcription. All right. Now, mRNA goes out and makes proteins. So mRNA now has like the code. It's like the code of how to make a protein.
Mm-hmm.
So this change from RNA to protein, this is called translation. See that? Translation. Right? So these are two words, from DNA to RNA, called transcription. From RNA to proteins, this is called translation. Right?
Mm-hmm.
So this is kind of showing a little bit more, you know, kind of what's happening, right? So over here, up in the nucleus. Any question?
How does it become an RNA? Like, how does the DNA become an RNA?
So see, do you remember how DNA was able to copy itself to form from one strand to do? So it's almost the exact same process. Take a look at this picture over here. You notice over here, so here's what happens. Let's suppose there's a certain gene that we need to code for. All right, so we decide, hey, we need to make more of this kind of protein. So the polymerase goes around and searches for that gene in the DNA. All right. So when it finds it, what it does is that it kind of temporarily opens up part of the DNA. Do you see over here how the double-stranded of the DNA has kind of opened up a little bit, and there's kind of like this empty space in the middle?
Mm-hmm.
All right. So what happens there is that then the RNA polymerase is just a protein that comes and helps along, copies a portion of this DNA, but this time it's not using DNA molecules. It's going to use RNA molecules, which are just slightly different, right? Ribose versus deoxyribose. It uses uracil and not thymine, right? So slightly different. So instead of copying like another strain of DNA out of it, it just copies a portion of it into RNA, right? So when it opens it up, right? Let's suppose let's suppose there's an A here in the DNA. Well, then it's going to fit a T. Let's say the next one is a C. So it's going to fit a G, and so on, right? Remember that same kind of idea for copying DNA?
Brother.
That same kind of idea is going to be used here, except we're going to copy it into RNA. Yeah, question.
Really? The DNA that is copied through transcription is copied into a messenger RNA?
Correct, messenger RNA. You see this? It says mRNA, right? mRNA, messenger RNA. Right. So this copy that's made is called mRNA. Right.
Mhm.
So now the mRNA goes outside over here, right? It goes outside, floats around. It finds a ribosome, right? And goes through the ribosome, right? When it goes through the ribosome, remember the transfer RNA that's holding amino acids?
Mhm.
They come in, and if they recognize, right, like this group of 3 base pairs here. Okay, right. Okay, you can sit over there. Right. So when the mRNA comes through the ribosomes, that's a good place for the transfer RNA to, if it identifies like a certain sequence. Let's say I identify a specific sequence. It can show up here, lodge in over here, and cause this amino acid to join this protein chain over here.
Mhm.
Right. All right, so let's take a look at over here, right? So here we have, so let's go back to transcription, right? So in transcription, here's what happens, right? Right, we took a look at this in the previous picture, right? You see this DNA kind of opens up a little bit, right? If we decide, hey, we need to copy this, part into some RNA. So what's going to happen? It's going to open up a little bit, right? The DNA is going to open up, and then this thing called RNA polymerase is going to show up, right? It's going to show up, and it's going to start to copy, the DNA into RNA, right?
Mhm.
So where should this, copying begin? There's certain, places called promoters in the DNA. Okay, so there's specific places in the DNA that have like a specific code that the RNA polymerase recognizes. Hey, I should start over here. So it starts over there at these places called promoters and starts making the mRNA right over there.
Mhm.
Any questions about this so far? Do you guys kind of get the general idea?
So brother, so basically what's happening here is, right, the during transcription, right, brother, the DNA is, some part of the DNA is copied into RNA by the enzyme RNA polymerase, right, brother?
Right. So this.
And then
Mhm.
that RNA is used to, or that mRNA is used in translation to make proteins.
Correct. Right. So right now we're making the mRNA. So once this is done, right now the only difference, right, of course, if you have A over here, right, in the DNA, you're not going to put a T, you're going to put a U because this is RNA. So in the RNA, you're not going to find any T's. Wherever you would find a T, now you're going to find a U.
Mm.
Right? So right now we're making mRNA, right? Now when mRNA is done, right, this is just another picture that's showing, right? So you see this site called a promoter site in the DNA. So that tells the RNA polymerase, hey, start making some RNA right over here. And so it starts making some RNA. And then when it's done, the RNA goes free. You see this RNA blue over here at the top? It goes free.
Mhm.
And it can go make some proteins with it. Right?
Mhm.
All right. So how does the genetic code work? Right? So we have this RNA, right? We have this mRNA. Now the RNA has different sequences, right? A, U, G, T, no, not T, U, C, G, right? Whatever, right? The mRNA now has a certain sequence of all these bases, right?
Mhm.
Right? So over here, right? So here's what happens, right? So each of those, each set of 3, right, codes for a specific amino acid. So let's suppose in the mRNA we had A, right? A.
Brother, that's
A.
the coding chart, right?
Yeah, this is a coding chart, right? So let's suppose in the mRNA we had this A, right? A, right? If we had this, that means that codes for the amino acid lysine, right? All these names here, these are the names of amino acids, right? If you put a whole bunch of these together, you make proteins.
Brother, that's 2 pairs of 3, right? Not.
Hmm?
That's 2 pairs of 3 for one protein, right?
Correct.
Sorry,
Right?
for one.
So take a look here. A can make a lysine. Also, AAG can also make a lysine.
Oh, both can make a lysine.
Both make a
Alright.
lysine. Look at this one for proline, right? For any of the, if you have any of these four, you're gonna put protein in the protein, or proline in the protein, right? CCU, CCC, any of these, right? You see, there's four of these combinations. If you have any of these four, the transfer RNA is going to come in and put proline into the protein. Does that make sense?
Yeah.
Right? So this one over here, methionine, is the start codon. So basically, the ribosomal RNA, the ribosome is reading this mRNA, and wherever you see AUG, right? AUG, that's where the protein starts. So most proteins, they always start with this thing called methionine. See this amino acid called methionine?
Yeah.
Most proteins, they start with this one. Now later on, the proteins are modified, so it might get cut off and stuff like that, right? But it pretty much always starts with methionine. Right? So this is kind of showing like a close-up picture, right? So you see this mRNA strand coming through, right? It's coming through the ribosome. And so let's say there's the AUG, right? Well, all of a sudden, this this transfer RNA is going to recognize that, and it's going to stick methionine at the beginning. And then you see the next three. You see this one over here has recognized, oh, the next three are mine. So it comes in and attaches. And you see this red one, this red amino acid, it's going to be attached to the blue, right? And so every time the mRNA, goes through a little bit, the transfer, you see these transfer RNAs that are kind of floating around? One of them is going to recognize, hey, that's my sequence. I'm going to go over there and I'm going to go put my amino acid in its place. Does that kind of make sense?
Yeah.
So every three, right? So you have 3 amino acids, right? So transfer RNAs are coming in and out. Most of them say, oh, I don't recognize that, and they leave. But whichever is the correct one, it says, oh, I recognize that sequence. It goes, attaches, sticks its amino acid onto the protein. Then the mRNA moves on a little bit more. The transfer RNAs, whichever recognizes, comes and sits there, attaches its amino acid, and so on. And this is showing the same kind of thing.
My brother, can you explain what promoters exactly are again?
Yeah. Just a second. So let's finish this, and then we'll go back.
All
Right. And I think this is basically kind of saying the same kind of thing, right? See all these transfer RNAs coming in, and they're attaching their own amino acid, right? Each one is going to come in, attach their own amino acid to this chain that's forming. So now you can have like this protein chain that's going to form here, right? All right, and I think that's it. So we'll stop there. If you have any, because I think the time is already up. So if you have questions, then you can stay. But otherwise, you guys can go to the next class. All right. So who had a question? Someone had a question?
Promoters.
All right, promoters. So, sorry.
Is the end of the lesson?
Yeah, it's gonna be the lesson.
Sorry.
All right, so you notice over here the promoter, right? So this is transcription, right? What does transcription mean?
Transcription is when the RNA, the DNA makes like a copy of RNA, right?
Right. So we're making a copy of RNA. So you see this RNA polymerase, this big blue protein?
Oh, yeah.
Right. You see this big, this big like a mouth-like thing, right?
The RNA polymerase.
You see this big blue blob. In
Oh,
the
this is a protein. All right.
That's a big protein, right? You see this one down here, same thing, protein. So this one's floating around, all right, and it's reading the DNA. It's looking at the DNA, and so when it reaches a certain site called a promoter site, it recognizes, hey, let me start right here. So then it starts making the RNA. So the promoter site.
Promoters are like there already.
Yeah, promoters are written into the DNA.
Ah.
Yeah, so promoter is like a sequence in the DNA.
Oh.
It's part of the DNA. So
cool.
it's a place where the polymerase recognizes, hey, let me begin here. And so whenever it reaches there, it recognizes, hey, this is where I'm supposed to begin. And so at that point, it opens it up and starts making the RNA. And
Okay, I understand
there's also like this terminator. You see at the end, there's something called terminator.
Terminator, yes.
So when it
That's
reaches-
like another. That's like promoter, but it's different.
Yeah, it's like same idea, right? It recognizes, oh, hey, I should stop here, so it stops.
So